We are here this week to know everything what TOC means and keep that in our brains, but we are adding a new piece onto that. We are adding endotoxins on it, and I think that is very important. I think it will strengthen our relationships with the customers specifically, because one of the things that I always remember in one of the big customers that I entered was GSK in my home country. When you enter there, I remember there was a big panel about different parameters they were measuring: TOC, conductivity, bioburden, endotoxins, particle counting. As you look at it, we are now already supplying 3 of them with the endotoxins parameter we will launch. So I see it as strengthening it, but also building more closer relationships and partnerships with our key customers, and I think that's very important, because maybe you never know, we can add the next 2 parameters to it, and then you have maybe the black box of measuring all the parameters and being one single source supplier to those big and smaller customers of us, and I think that would be great.
Our Eclipse platform is the exact same test that is used on a 96 well plate. It is a compendial assay. We have built our platform and our pre-deposited endotoxin standards to behave more appropriately to get better curves than on a standard assay. We're able to do exactly as many samples as a 96 well plate right now, with much less volume of sample and much less volume of reagent.
We're able to cut horseshoe crab lysate usage 90%. Which is huge, because that's also the most expensive part of the assay outside of labor.
We also are able to use centrifugal technology, and that allows us to have so many samples that we don't lose any efficiency from the 96 well plate. I'm excited to be part of this brilliant team of engineers. My part was, I'm the biochemist responsible for the deposition and positive product control pre-deposited standards. That is what allows us to have a self-contained liquid handling device on a microfluidic platform that is the most dense of any microfluidic platform out there.
So if you look at the actual plates itself, we're beyond the limit of microfluidics in terms of the spacing, the density. Look at anything else out there where they call it microfluidics. It's not. This is microfluidics on a huge platform, something that's never been done. Again, something that most said, we can't do that, it can't be done. You should go back to the drawing board and figure out something else. We kept scaling it up, and each time we had those challenges. We had to have, back and forth reviews globally. These design people, they had all sorts of modeling. They would put it all in the computer, model up the fluidics, model it up, spinning it at different rates, and send us a design, and we manufacture that, and then tweak it back around. All of those microfluidics have to move at exact precision, frequencies, velocities, otherwise it doesn't work. Everything you see in there looks really simple when that's working, but it has to operate precisely at every step, every moment. You can take a video of these little 55 micron channels of liquid moving around in there. It all happens almost instantaneously. It's almost magic if you look at the design, how it has to operate to the end result, and it makes it look super simple with a ton of work in the background. The true beauty of this product is taking a huge amount of complexity, simplifying it down. It looks like something that isn't that complicated.
That's a highlight of this product. Again, at the end of the day, we're taking a very complex assay and we're making it so simple anybody could run it. No lab experience, no scientific background or education, and you can run this assay.
If you look at it when it's operating, there's a lot of stuff you wouldn't even imagine. It kind of operates like a washing machine, but really at a high-tech level.
That's one of the great things about it, is you can take somebody that has no experience and they can run an endotoxin assay. And so when we're in the selling process, you can take the CEO or the CFO or somebody in a suit that's never been in a lab, and all you have to train them is how to pipette.
The customer, when they see it, they use it once, it's completely obvious that it's the most simple thing in the world, but there's like 100 things that go in the background that had to work, that are difficult to make work. It just seamlessly operates without them knowing anything about that.
The ability for a user to take this piece of plastic, inject some liquid into it a couple times, hit go, and this thing to make many, many measurements very quickly is really impressive to me.
That's revolutionary. That's never been done in an endotoxin assay today. And again, I think it continues our legacy as the Sievers product line. We've brought simplicity to the TOC analyzer. You press the green button, you get a TOC result. In this instance, we're taking 242 pipetting steps, 1 hour of person time of labor to produce and set up an assay. And we've created 27 pipetting steps in 5 minutes. It's unheard of. It doesn't exist today except for right here in our building, and we're going to show you that this week.
Our Eclipse system makes adding samples infinitely easier. It removes operator-to-operator variance. It has the potential to be run in series with multiple instruments. We're also able to minimize the operator time by having pre-deposited positive product controls on our plates. Additionally, we have pre-deposited standards, so the customer no longer needs to purchase a controlled standard endotoxin or the reference standard endotoxin. We also have more negative controls, giving the customers better visibility on their source water.
Their efficiency now is improved greatly, so they're spending their time focusing on other aspects of their QC lab where they can divert those resources. So we're providing them an opportunity to tremendously improve their efficiency within the lab. We're ticking every box that we're told you need to tick. It's validatable. That is huge.
There's a lot of moving parts physically and metaphorically that come together to make this thing great.
And to have 3 out of the 4 major ultra pure water tests under our portfolio and to be compendial and to have the best technology is an extremely strong value proposition, and one that I'm just fired up to be a part of and can't wait to see what happens in 2019.